It's a great pleasure for me and my colleagues to visit Ho Chi Minh City and Vietnam University. You have shown us in the few days we've been here wonderful hospitality. We've been able to meet with your faculty and administrators, and now we look forward to meeting with students as well. Our goal is to continue and strengthen the ties between Vietnam University and UCLA, and our particular interest and goal has to do with the mentoring of young scientists to set young scientists on the path to successful scientific discoveries. So my talk this morning involves just a small part of what goes on at UCLA, a huge university like your university. Our university includes schools of basic science, engineering, medicine, but also law and history, humanities, social sciences. So I'm speaking now of the work of only one laboratory out of probably 500 laboratories at UCLA. And my laboratory has been concerned with the amyloid state of proteins and also the connection of the amyloid state of proteins to diseases, and most particularly cancer. So I will attempt in the next few minutes to answer 4 scientific questions. And the first is, what is the amyloid state? Second, what's the connection of amyloid fibers? You can see in the background here the long fibers, which are protein fibers seen in the electron microscope. Those are amyloid fibers. What's the connection of these fibers to diseases, including cancer? What are the atomic structures of these amyloid fibers and of smaller units of the same proteins that are called oligomers? I'll just touch on that topic. And finally, can these agents of disease, these causes of disease, can they be inhibited, including in cancer? So let me ask you, can you hear me in the back? Raise your hand if you can hear me. Yes. Raise your hand now if you cannot hear me. In the back, can you see the screen? If you can see the slides on the screen, raise your hand. Thank you. So first of all, what is amyloid? Well, they're protein fibers that are unbranched and elongated. And on the left-hand side, you see some of these fibers. They differ a bit in width, but you can see them in the electron microscope. Pathologists who first discovered them in samples from the brains of patients, they define them as unbranched, elongated fibers associated with disease, and that they bound a dye called Congo red, which fluoresces when seen in the microscope. That's not a very satisfying definition to a biophysical scientist. So the biophysicists now define amyloid as fibers which give a cross beta diffraction pattern, and that's shown here. If you shine a beam of X-rays on a fiber, then you see a pattern on film, or the X-ray detector, which shows what we call a reflection at 4.8 angstroms from the center, and that shows that these fibers have individual molecules which are elongated and separated by 4.8 angstroms. Also, at right angles to that, there's another reflection at 10 angstroms resolution, and that shows that these fibers are organized into sheets, and that the sheets are about 10 angstroms apart. So now that's the definition which is used by biophysicists. And also another property. If you take the protein, which is going to form an amyloid fiber, and you take the purified protein and put it into solution and wait for a time, it does not immediately form the fibers. You have to wait for hours, sometimes days, and then suddenly it forms fibers. But if you take a pre-formed fiber, one that is already formed, and put it into the solution, then it seeds fibers and they form immediately. So this shows that there needs to be an instruction, a pattern for forming the fibers that takes time to form on its own, but which can be provided by the seed of the fiber. So here I list some of the many diseases associated with these fibers. And each of these diseases, and also 4 times this number, pathologists have found in samples from patients that there are the same sort of fibers associated with that disease. Usually, it's a particular protein associated with each disease. In the case of Alzheimer's disease, there are 2 proteins. One extracellular called A-beta, a short peptid of about 42 residues, and an intracellular protein, which form fibers. The fibers which you see here are A-beta fibers. Also, in diabetes type 2, many other diseases, some of these diseases are transmissible from animal to animal, cell to cell. Those are the so-called prion diseases. They're amyloid diseases, but transmissible from organism to organism. And then there's still other diseases which pathologists don't yet classify as amyloid diseases, but biophysicists do. Those include Parkinson's disease, ALS, and now many cancers. In some cases, it seems clear that the fibers cause the disease. That includes these diseases like transthyretin amyloidosis or light chain amyloidosis. In those diseases, fibers form in us, in the patient, and they form so many fibers that it clogs our organs until they fail: heart, liver, kidney, lung. So in those cases, it's the fibers that are causing disease. That also seems to be true in the case of cancer, where it's the protein P53 which forms the fibers. In other cases, mainly the diseases of the brain, like Alzheimer's, Parkinson's, ALS, prion diseases, in that case, it's smaller units of the same proteins. Those are called toxic oligomers. I'm just going to mention them briefly at the conclusion of this short talk. So why study these diseases? The reason is that they are a tremendous social burden to our societies. In the United States, the last year for which there are accurate records, there were 5.5 million patients, and it's about 26 million patients with Alzheimer's disease worldwide. By mid-century, it's expected in the United States alone that there may be as many as 20 million patients. The cost of care for Alzheimer's patients in the United States in 2010 was 183 times 10 to the 9th billion dollars. It's estimated that by mid-century it will be 10 to the 12th dollars, $1 trillion a year, which is a significant fraction of the gross domestic product of the United States. Something must be done to halt these diseases, or our societies will be spending all our efforts, all our money in treating patients. In addition to the cost, in Alzheimer's disease, there are usually 2 family members who spend their time caring for patients unpaid. Diabetes type 2, another amyloid disease, almost as many patients, almost the same economic burden to society. Parkinson's disease is an order of magnitude smaller, and cancers, there's a large number of cancers, that are caused by amyloid. The number is not yet known, but together these diseases represent a great problem for society. That's why every one of you should be working on these diseases. So the work in our laboratory has gone on for 12 years, and it's ended up with two hypotheses. They're sort of the end of what we've come up with. For the fibers, which I've mentioned, we believe that the fibers are caused not by the complete unwinding of a protein and then rewinding into the fiber. Rather, the protein just opens to expose a short segment of about 6 amino acid residues. And this segment is self-complementary. It binds strongly to the same segment in other molecules. And that's what causes the fiber, a short adhesive segment. And this segment forms 2 beta sheets, like I showed you in the first slide in defining amyloid fibers. And we call that structure, which I'm going to show you, a steric zipper. And these steric zippers can be inhibited. We can design inhibitors and stop the formation of fibers. That's the first hypothesis. The second one we call the cylindrin hypothesis. This deals with the toxic oligomers. Same proteins, but instead of having millions of molecules in the fiber, the toxic oligomers have a much smaller number, maybe 6 to 20 molecules, and those are the toxic entities in the brain diseases. And our hypothesis is that they form a different sort of beta sheet, which I can show you, anti-parallel, out of register, twisted into a cylinder, which we call a cylindrin. So here's a picture of a steric zipper, and you can see the extended protein chains are stacked up. Here there are only 5 chains. In an actual fiber, there are about 50,000 to 100,000. So you have to imagine that this fiber, which goes in that vertical direction, has 50 or 100,000 chains. And you can see that each chain is extended and each member is stacked exactly above or below the one below. That's what we call in register. And if you look down the axis of the fiber, like there, you see that the side chains of the short segment, they're interdigitated like the fingers of your 2 hands. And it's that interdigitation that gives the stability to the amyloid fibers. So from now on, I'm just going to show you that view down the axis. Here we are looking down the axis of an amyloid fiber of a short, this is the short adhesive segment, only 7 amino acid residues. And you have to imagine that there are 50,000 layers of this, 50 to 100,000 layers, and you can see that the amino acid side chains are interdigitated. That's what holds the 2 sheets together and makes it so stable. Once a structure like this starts to form in your brain, it's not going to dissolve. If we take this particular fiber, the only way we can dissolve it is in 1 normal sodium hydroxide or in pure formic acid. These are very stable structures once they form. So in our laboratory, we now have determined structures for about 120 different short segments from the disease proteins in many different diseases. And I'm not going to have an opportunity to describe all of these, but I will mention the one from P53 related to cancer, and I will mention the one from tau of Alzheimer's disease. But there are many others shown here, always looking down the axis of the fiber. How do we identify these short segments that form the amyloid fiber? Well, we use a computational or bioinformatic procedure. We take the structure that we know. Here we are looking down the 2 sheets. You can see the side chains which are interdigitated. We remove those side chains from the structure, just leaving the backbone of the fiber. Now we scan through a sequence of a protein. This protein happens to be A beta of Alzheimer's disease. And at each position of 6 residues, we take the side chains of those 6 residues and put them on the backbone here. And we ask, does it form a steric zipper? And the way we ask that is by looking at the energy. And if the energy is below a threshold that we've established empirically, this threshold you can see here, this shows the energy for each 6 residue segment as we scan through the sequence. And for A beta, you see that there are 1, 2, 3 segments that will form a steric zipper. When we synthesize these 6 residue segments from A beta, just 6 residues, they will form a fiber. And they also form small crystals that allow us to determine the structure. So this shows the amyloid fibers from just short segments, 7 residues, even 4 residues. And here are the crystals. You can see the crystals are exceedingly small. This whole bundle of crystals is 0.5 millimeter across. In the electron microscope, we can see the crystals. They look like good crystals. They have edges and faces. So we knew that we could determine this structure if we could learn how to work with such small crystals. Well, that took 4.5 years, but we were able to do it. And this shows, this on the left-hand side, you see 100 crystals from which we determined the first structure. And they are scaled by the smallest American coin, a dime, which is about 1 centimeter across. Here's the dime, and here are the crystals. And I've enlarged 4 of them here. And what you can see, most of what is shown is just the glass pin. This is the crystal, this tiny thing on top. So these crystals are just 2 micrometers in cross-section and up to about 50 micrometers long, but usually much shorter. But that was enough we learned to determine structures. I won't go into details beyond that. So what about inhibiting these fibers? Well, we learned how to inhibit them, and so far we've done that for 3 different types of fibers. I'm going to start with tau and then move to P53. Tau is one of the Alzheimer's proteins. So first of all, we use our algorithm to identify a short segment, which is the adhesive segment, which causes the proteins to aggregate into fibers. Here it is, 6 residues. We call it, we just pronounce the sequence, Viquidic, think of that as a word. So we synthesize that sequence, and sure enough, just those 6 residues form a fiber that looks like the fiber of the whole protein. And those 6 residues form microcrystals again. We can use those microcrystals to determine the structure. The structure is 2-sheet steric zipper, as it always is. Here's the axis of the fiber in the steric zipper. And now that we know the structure, we can design a peptide which sits on top of the steric zipper and prevents another layer from forming. So that's an inhibitor. And in this case, we designed an all-D amino acid peptide. And if we take this inhibitor and put it in a solution of the protein tau, it completely prevents fibers from forming. That's shown. This is the process of design. So you see 1 sheet of the steric zipper, and now in the computer we try all possible combinations of amino acids, looking for ones that bind tightly to the steric zipper below, and then we allow the structure to anneal to find its lowest energy state, and then we have another round of looking for the best amino acid side chains, and then another round of annealing. This is an iterative process, and at length we come up with several candidates for inhibitors, and then we test them and find the best one. So this shows our best inhibitor for tau. It's an all-D amino acid, 6 residues with this sequence, and this shows the formation of fibers using fluorescence against time. And if we put tau in solution, this is the protein tau, and we shake it, after about 30 hours, it forms fibers. But if we have the inhibitor present, we can shake it for a whole week and no fibers. So the inhibitor really works. Does the inhibitor work the way we designed it? Well, that was a question of the referee of the Nature paper, so we had to prove that. And graduate student Howard Chang took this inhibitor and he made a gold adduct. We could see in the electron microscope, and we could see that the inhibitor went on the ends of the fibers as it was designed. And the referee said, "That's good enough for me, you can publish your paper." So now I move to my last topic, P53. So P53 is involved in most cancers in one way or another. It's called the guardian of the genome, involved in many cellular processes, including repression of the proliferation of cells. So if P53 is gone, you're in trouble. Now your cells can proliferate. So it was shown in 2011 in a paper in Nature Chemical Biology by Xu et al. That P53 mutants form amyloid aggregates in cells, and also they found that this was true in samples from prostate cancer. And if you have a P53 mutation in one allele, and it forms the amyloid, then it pulls the wild-type P53 into the amyloid too. So it's a dominant effect, a gain-of-function mutation. Also in the same year, in another paper, it was shown that mutant P53 forms amyloid aggregates in breast cancer cases. And since then, it's been found in ovarian cancer and other cancers as well. And this shows a picture of it. Imagine that this is P53. Here's the short segment in red, which is the adhesive segment, which forms the amyloid. If there's a mutation anywhere, it destabilizes P53. That opens the protein so that the short segment is exposed, and now it aggregates and pulls in the wild type as well. So now your cells have no active P53, and they are free to proliferate. So what did we do to design an inhibitor? Exactly the same process that we used with tau. So we identified at the top the segment, which is the adhesive segment in P53. It has the sequence L titles. We synthesized that segment, 7 amino acids. It forms fibers. It forms microcrystals. We can determine the structure. Actually, we determined 2 different structures. To each of these, we designed inhibitors. The inhibitors designed to this structure didn't work. The inhibitors designed to this structure did work. Here we show the formation of fibers of P53 in vitro as we add more and more inhibitor. When we get one inhibitor to one P53 molecule, fibers are very strongly suppressed. They don't form. At this point, we made a cell-penetrating version of this peptide. And that's shown up here. We just add a polyarginine tail to the peptide inhibitor. And now, when we add that to cancer cells, it allows P53 it prevents it from going into the amyloid, and those cells now have their P53 reactivated, and it kills the cells which are proliferating. So that was very encouraging, and the next step was to try it in mice. And the nice thing about UCLA, we have lots of experts in our cancer center, and we worked with Sanez Memrazda in her laboratory, and she has mice in which she implants cancer cells, ovarian cancer cells, in the hind legs of mice, and in 2 weeks, tumors grow. This shows the tumors growing. if we inject just the buffer that we use, doesn't prevent tumors from being grown. If we inject our inhibitor, look at this. The tumors shrink after just 2 weeks of injecting our inhibitor to the mice. And if we scramble the sequence of the inhibitor so that it shouldn't work, it doesn't work. So we believe that our inhibitor is inhibiting the growth of these these tumors in mice. And then very briefly, I'll just end by showing you what we believe is the structure of the small toxic oligomers that form in Alzheimer's and other brain diseases. And it was graduate student Arthur Laganowski, now a postdoc at Oxford University. He wanted to grow a crystal of a small oligomer, and you can see he did. These are his 96 well trays. These are his 24 well trays. It took him 140,000 drops. Well, he had help from a robot that made the drops, but these were his ideas. And at last he grew a crystal so we could determine the structure. And here is the structure. It's not a steric zipper, it's a cylinder. And you can see the strands are not parallel, but they're anti-parallel. And they form this small cylinder. And if you look down the cylinder, it appears to be hollow, but it's not. If we put in the space-filling atoms, it's not hollow. And if we open it, you may be able to see that the strands are not in register. Each pair of strands slides up compared to the others so that the cylinder can form. So we believe this is the nature of the small oligomers. So just to summarize here, 12 years of work by lots of scientists, we believe that the hypothesis of the steric zipper is well supported because we can use that hypothesis to inhibit the formation of fibers. With the cylindrin hypothesis, we don't yet know that we can inhibit cylindrins, but that's what we're working on in our research group. And just to show you the photographs of some of the people who are involved, this work involved at least 20 scientists over 12 years, most of them for only 4 years or so. The first crystals were grown by Melinda Balburney. She now is pregnant with her 4th child. Rebecca Nelson has 2 children. She determined the first structure, and Michael Savoya is our great crystallographer who has been involved in all of the structures, and the small oligomer structure was determined by Art Laganowski, and the design of our inhibitors was done by Stuart Sievers for tau and Lin Jiang for P53. So I thank you for your attention, and I guess this afternoon we can have questions about it.